Hi, this lecture will go over the first set of biochemical tests that we'll be doing. Your classmates will be starting this Tuesday evening in lab, and we will finish this up on Thursday. This can be found on page 46 and 47 in your course pack. So in order to identify bacteria, we've already talked about several of these things. We look at the cell morphology, that is the shape and arrangement, and the gram stain characteristics. We can look at the colony characteristics, that is what it looks like growing on a plate, and I'll be sending that to you. And then other culture characteristics. Does it grow better at one temperature or another? Does it maybe produce different colors? What does it look like? Again, I'll be sending you photos that will help you determine if there are any particular characteristics about your bacteria. But that's not enough to help us identify a bacteria. So if we've isolated some bacteria from a patient's throat and we found it to be a gram-negative Diplococcus, the colonies are white, raised, entire, and they grow best at 37 degrees. But is the bacterium Neisseria subflava or Neisseria meningitidis? Neisseria subflava is a common flora in humans. It does not cause disease, at least not normally. However, Neisseria meningitidis causes the illness meningitis, which can be very serious and in fact can be fatal. So we'd want to know which bacteria we're looking at. One of the things we do is use biochemical reactions. It's very useful for identifying bacteria, and this is done often in medical labs. The biochemical reactions are often mediated by enzymes. That is, an enzyme helps the bacteria to perform a certain biochemical reaction. And some of these reactions relate to the pathogenicity of the microbe. For instance, if a pathogen can form hemolysis, that is, break down red blood cells, it can be more pathogenic, make you sicker. If a bacteria has DNase activity, produces DNase that can break down DNA, again, that's a virulence factor that can make this microbe more pathogenic. So the absence or presence of these enzymes can differ at the species and even the strain level, but they can be determined by doing biochemical tests, and that's what we'll be doing in the next couple of weeks. Specifically in this lab, we'll be looking at the fermentation of carbohydrates, and we'll use 4 different sugars. Can this bacteria, your pet, ferment glucose, maltose, lactose, or sucrose? And if it can, there can be 2 end results: the production of acids and or the production of CO2. So we'll do a test to look at that. We'll do the Simmons citrate test. That is, the bacteria can use citrate as its sole carbon source, and it can catabolize that citrate. If it does, then the pH rises, and we can tell the difference by a color change in the media. Does your bacteria produce indole? That is, can it break down tryptophan to produce indole? And can your bacteria carry on nitrate reduction? Can nitrate be reduced? And that also will cause a color change. And we'll also look at hydrogen sulfide production. If your bacteria can break down cysteine, it produces hydrogen sulfide, and we see a color change. And then we will also look at motility. That is, does your bacteria, is it motile? Can it move? Does it have flagella or other cellular components that help it move? So the fermentation of carbohydrates. As I said, this detects the ability to ferment a particular carbohydrate. We will look specifically at glucose, sucrose, maltose, and lactose, but there's a whole host of other carbohydrates that can also be tested. And we're looking for the end products of fermentation. That is, can this bacteria produce gas in fermentation? And if so, it will collect in a little tube in our tubed media. I'll show you that in another photo. And if it produces acid as an end product of fermentation, we'll have a color change. This media uses phenol red, and phenol red is an indicator, a pH indicator. So with With no acid production, the phenol red tube will be red. But if acid is produced, then the pH drops and the phenol red indicator will turn yellow. The citrate test. As I said, this determines whether an organism can utilize citrate as its only source of carbon and energy. If it does, the catabolism of citrate produces a basic product, so the pH rises. And in this case, this media has bromethymol blue as a pH indicator, and as the pH rises, the tube will turn blue. Indole. Can your bacteria produce indole from tryptophan? So can it break down tryptophan? If it does, we will add a Kovacs reagent to that broth that the media is growing in, and if it produces indole, we'll get this nice bright red ring that you see in the photo on the left. The photo on the right is negative for indole production. And can your bacteria reduce nitrate? So this test will detect the ability to reduce nitrate ions to nitrite ions, in which case your bacteria has the enzyme nitrate reductase, or can it produce nitrogen gas? That is, it reduces the nitrogen all the way to nitrogen gas. So from nitrate to nitrite to nitrogen gas, and we look at color changes. And then also SIM, the hydrogen sulfide formation. Does your bacteria produce the enzyme cysteine disulfurase? If it does, cysteine can be broken down to produce hydrogen sulfide, and in our media, there's some iron in there that in the presence of hydrogen sulfide will produce iron sulfide and will have a black precipitate. And we'll look at motility. This determines whether the bacterium is motile. In the photo on the right, you can see where the bacteria was stabbed. We use a needle to just stab this bacteria into a semi-solid tube of media. And if the bacteria is motile, you can see it spreading out. You can see in the photo that the entire tube begins to get hazy because the bacteria is spreading. In the tube on the right, the bacteria is only growing where it was stabbed into the media. So we don't get any motility. The bacteria stays local to where it was stabbed into the media. Here's the tubed media that we're going to be using that we'll be inoculating. So inoculating means to add some bacteria to this our clear media that has nothing growing in it. The 4 tubes on the left are those phenol red tubes that we'll use for fermentation. The 4 tubes each one contains a different carbohydrate. And you can also see that other inner tube, the Durham tube, that's coming out of the media. Right now, those tubes are filled with the phenol red broth base, but as we inoculate the bacteria into those tubes, they may or may not turn the media yellow if it produces acid from fermentation. And if it produces CO2 gas, that gas will be collected in those little inner tubes, and we will see the media being displaced, and you'll just see a clear area in the tubes. And again, you'll have pictures of the results of your bacteria next week. The next tube, the second from the right, is just that nitrate broth. Again, all I'm showing you here is that an uninoculated tube is just clear. There's nothing growing in it. And then that tube on the far right is the Simmons citrate. Right now you can see that that's green. No bacteria has been inoculated there, but when we inoculate your bacteria, if we get that blue color change, it will be because your bacteria can catabolize citrate as its energy source. The pH will rise and the green media will turn blue. Here's how you'll be graded on this. So each biochemical test should have 3 parts, and this also is listed in your course pack. So the first part is to describe or observe what you saw, or what you'll see in your photographs. For instance, the media turned blue. Then the interpretation. Well, why did the media turn blue? The color change is caused by a rise or a change in the pH. And then what is the conclusion? So in this case, it's a positive result indicating that my bacteria can utilize citrate as the sole carbon source. So again, each observation and interpretation has three parts. What you observed, what the interpretation of that is, and then the conclusion. For those of us in the lab, here's what we'll be doing and what I'll be doing for you on Tuesday evening. I'll be inoculating the tubes of media with your pet bacterium. And then we wait either 24 or 48 hours to make our observations, right? The bacteria needs time to grow. We need to look at fermentation results within 24 hours. So I'll inoculate your tubes on Tuesday evening, but on Wednesday I'll come back and observe those fermentation tubes, and I'll take photos. Other tubes take a little bit longer, so we'll be doing those on Thursday evening.